Thank you, and thank you to the organizers for organizing this fantastic conference. I guess the organizers wanted me to present something that's in between the previous two speakers because there's a very wide range to cover. But also I think probably what I need to do is like combine hummus with black holes somehow. Okay, so the mission of my lab, the Neural Interface Engineering Lab, is to take an emerging toolbox, a very strong neurophotonics toolbox that allows us both to see, to image neural activity with cellular resolution, with the full dynamic content of neural activity using light sensitive calcium sensors or other functional sensors. And a second part of the toolbox is actuators, or like an arm here, which are based on light sensitive ion channels and related tools, often called optogenetics. And we take these two complementary types of technologies and put them together to form bidirectional neural interfaces. We can do this at the microscopic resolution by combining, for example, two-photon microscopy together with holographic optogenetics. And we can do that with single cells across distributed networks. We can do this on a mesoscopic scale by combining technologies like optoacoustic imaging together with ultrasonic neuromodulation, which I will explain later. And in the lab, we sometimes also do things that combine across these different scales. It's kind of an emerging field. So let me give you a few examples of what we do and how we do this. So when I moved to NYU, I actually came for sabbatical to work with olfaction researcher Dima Rinberg. And we put together a system that combined GCamp calcium indicators that allow us to image activity, again, on cellular scale, together with Crimson, an optogenetic tool that allowed us separately to control activity in this cellular population. We did this in the olfactory bulb, so we can both image and control activity, and we can do this in awake behaving mice that are head fixed, but they can do full behavioral tasks and can be trained in days, weeks, and basically we can do very sophisticated behaviors, which is something that Dima has developed. And a few words about the system in which we do this. In olfaction, rodent olfaction, odorants arrive and land on olfactory sensory receptors in the olfactory epithelium. The signals then propagate to a relay station, but a very sophisticated relay station in the olfactory bulb. And from there, they continue to areas of the brain. The sensory neurons, the olfactory sensory neurons have receptors on them that are sensitive to specific molecules. And there is a labeled line, a molecularly specific communication to specific glomeruli in the olfactory bulb. And there are about 1,200 different such units in mice, a little less in humans. So individual glomeruli. The mitral cells are the output that sends information to cortex. And a single unit in this setup basically has something on the order of about 5,000 receptor cells, ultimately sending information through 20 mitral cells. So there's a lot of compression in the system as well. And when different odorants, say a banana or a rose, they excite different olfactory receptors, and this causes a different type of excitation pattern. So ultimately, what we have is basically a stimulus like a rose, eliciting a spatiotemporal pattern that we can visualize at the glomerular level. We can also visualize these responses, also another spatiotemporal pattern at the mitral cell layer, which is the output, as I've mentioned. And we can also basically train animals to report what it is that they're sensing. This system is highly accessible at this point, and we can not just only observe it, we can actually take these abstracted spatiotemporal patterns and we can generate them. For example, we could do this at the glomerular level using projectors, just like this type of projector that we're using here. At the mitral cell layer, we can use holographic optogenetics to activate single cells. And again, we can observe the behavior. So ultimately, what we can do is we can replace the real rose with a synthetic rose and still elicit perception. Okay? And these are the types of manipulations. We can also come with these tools, not just as replacement tools, but also as perturbation tools, and perturb natural perception. So keep that in mind. The tool of choice for a lot of this work is based on the generation of holograms. So Yoav already introduced SLM, spatial light modulators, but we use them in the opposite way. Instead of using them in the imaging path, we shine a laser in this, at the spatial light modulator. The wavefront interferes, and we can, by engineering the PSF, we can generate all kinds of patterns, and we can change them very rapidly in space-time. And both my lab and several other labs in the field have used this to create highly efficient and controllable cellular resolution patterns. We can even do this at a subcellular resolution, but for what we'll talk about right now, the cellular resolution is the most interesting one. And we've calibrated our systems for these in these experiments in vivo, using in vivo patch recordings, to bring them to the level where we generate single action potentials in single neurons. So basically controlling the dynamical process at its intrinsic resolution. And then we can train an animal to reliably detect the co-activation of groups of neurons. For example, and we've developed methods to do this, and basically the animal can, after a few days of that, can reliably detect a synthetic activation pattern of about 30 co-activated neurons. And then we can start dropping neurons and basically see what is the threshold of perception in which the animal basically can no longer detect this activity. And what we've found consistently is that this is on the order of 5 to 10 co-activated neurons. So a volley of about 5 to 10 action potentials across a small population of neurons. And we've done this in different ways, in different animals. And this is actually pretty remarkable because if you look at kind of the cacophony that arrives at cortex, it includes about 1 million, give or take, action potentials per second arriving from something like 50,000 mitral cells in the olfactory bulb spontaneously. On this background, the animal can detect a very small synchronized volley And then we can do other things. We can not only explore what is the number of co-activated neurons, we can look at what are the crucial features in this activity. And for example, we can check whether it's synchrony, for example, by taking our volley and desynchronizing it. What we've found is that even adding a jitter of about an extra 20 milliseconds to this volley basically eliminates the animal's ability to detect it. So synchrony is extremely important, but not everything is important. For example, if we take our volley and just shift it in time, and we're shifting it relative to the sniff pattern by about 70 milliseconds in this case, 100, up to 100 milliseconds, we see very negligible effects of that manipulation. So the arrival of a synchronized volley of action potential is super important. We can go beyond what is the threshold of perception, what is the threshold of detection for perception, by looking at things that are more like objects. And an object could be, let's use musical analogy. So if you take a tune, let's say, Hey Jude, you can start playing with the pitch and timing of individual notes. And at some point, it will no longer sound like Hey Jude. It will sound like something completely different. And so you can wonder what in our spatiotemporal pattern will have the same effect. And to approach this question, the Rinberg lab trained mice to basically detect one spatiotemporal pattern versus a multitude of other patterns, basically detecting a specific object in this case, and then ask the question of if you just probe this object randomly or in a structured way, when will it sound? When will it detect it, or when will it recognize it as a different object? And this has been published, so I'll just give you kind of a quick summary of the findings. Earlier spots are much more relevant than later spots. It's so-called primacy effect. It seems like the different spatial channels, the glomeruli in this case, are independent, and the animal is extremely sensitive to their relative timing, but very weakly sensitive to a shift of the entire pattern, which probably a lot of regular signal processing methods will also reproduce. And in fact, these are qualitative statements, but we can actually put together a whole host of manipulations that were done in these experiments into a quantitative spatiotemporal template matching model, basically a sort of matched filter for space-time patterns, and basically use that to calculate a perceptual distance from the object. And not showing it here, but our model really captures very well quantitatively a whole host of results in these experiments. So let me just summarize quickly this part of the talk. In New York, we really like our pizzas. When we see a pizza, when the sensory system sees a pizza, it basically excites a whole host of spatiotemporal patterns across multiple sensory systems. This is a process that we call encoding. And this process is very well studied and understood because we can manipulate the pizza in many structured ways. But it is the readout of this process, of these spatiotemporal activity patterns, that is crucial in order to form a perception of this pizza, which is ultimately what the organism uses in order to guide behavior. Okay, so pizza rat will not be able to do what it does without this internal representation of the pizza. And this is important not just for understanding how real neurons work, it's also important for artificial neural networks, which I know is very interesting for this community, because generally we have this decode phase there, but decoders, both in systems neuroscience and in artificial neuroscience as a result, essentially have a structure that is assumed rather than really obtained from real understanding of the nervous system, because only recently we have tools that can start probing this stage. And if to conclude the results of our early studies using these tools, what I can tell you is basically that just like a radiologist eye basically hones in on the lesion, the same way the brain has a system that hones in on synchronized activity patterns that arrive very quickly after a stimulus, after a sniff, after an eye movement, etc. How do we scale this up? Still, this is about mesoscopy. How do we scale up these tools to a larger scale? Because as we know now, although we have these beautiful tools that operate on less than a cubic millimeter, when we want to scale up to the macro scale, we have other completely different performance profiles. We have tools that only look at hemodynamics. They generally have poor spatial temporal resolution. And really what we want is to have this kind of full dynamic capture at the level of the entire organism, a little bit like imaging the whole Earth at a single person resolution. So really where we want to be here on this plot is in this area where we have cellular resolution, something akin to cellular resolution, and and the ability to capture dynamics directly on a larger scale. So how do we do this? I don't know. But we can come close to this using existing tools. And I've been collaborating for a decade now with Daniel Rosansky, who is now at ETH and the University of Zurich. And together, we set out to combine tools that could approach this kind of large-scale interfacing problem. We can use optoacoustics, which is the optical method that has the highest ability to penetrate into tissues, together with ultrasonic. So we use that for imaging, together with focused ultrasound for excitation. A few words about optoacoustics and optoacoustic tomography. Optoacoustics uses basically light to excite. When a short light pulse is absorbed in tissue, it generates ultrasonic waves. This is a very strong effect if you're using nanosecond or shorter pulses. And you can pick up this ultrasound on an array of ultrasonic receivers and basically back project it to generate an image. You can also do it model-based. I'm not going to talk about the many different ways and interesting mathematical aspects of doing that. But a single laser pulse, in this case, using this excitation methods, unlike our usual laser scanning methods, allows you to generate a full volumetric acquisition. You can actually quickly switch pulses between different parts of the spectrum and get resolutions that are mesoscopic on the order of about tens to hundreds of microns per pixel, per voxel. And you can do it across centimeter range brain objects and brains. So just to show you the spatial temporal capability of such techniques, for example, we can, because each pulse gives a full volume, we can image at 100 volumes per second dynamical processes like the swimming of this zebrafish, young zebrafish larva, or a beating heart. So this is results from the Rosansky lab. more interesting for us was the question of whether we can use this for calcium imaging. So we approached this question together using the state-of-the-art calcium imaging probes, which were actually developed originally for fluorescence, but it turns out that they also have an absorption change that is associated with this fluorescence change, as seen here. And as a result, we can measure a change in the optoacoustic signal. And in fact, we showed that in our early studies by looking at transparent organisms where we can see both fluorescence and optoacoustics simultaneously, and they both track as the calcium is changing. And we could do this also in scattering zebrafish, adult zebrafish brains. But really the most interesting was whether we can take it to our favorite organism of mice, maybe to humans at some point. To do this, we built setups where the mouse is placed under a water-filled vessel coupled with a membrane to its brain. And we showed that we can actually measure functional responses using this GCAM family of indicators. More recently, we've been looking at whether we can do this in the near infrared, and the reason is that with these indicators that were optimized for fluorescence, we can only image the top of the brain, the cortex, because light doesn't penetrate very deep. But if we move to infrared, we can look at the whole brain. And we partnered up with Robert Campbell, who developed the first family of near-infrared calcium indicators. And Sarah Shaykevich, who is familiar to many of you because she's a part of CBI as well, has been hard at work in her PhD to basically develop the methods for imaging optoacoustically these near-infrared calcium indicators, and we will hear more about it soon in her PhD defense. We can also image hemodynamics in the near-infrared. Blood does have a very strong and functional signature in near-infrared wavelengths. We do that together with calcium imaging, which is done fluorescently. And then we were also very happy recently that we were able to demonstrate that we can image other types of functional processes going on in the near-infrared, for example, by adding a near-infrared tracer to the CSF, we can image CSF flow dynamics. We can basically correlate what we see with fluorescence and what we see with optoacoustics across the whole brain. Basically, measure CSF flow, which is which is also related to the lymphatic flow, which is very interesting for aspects of neurodegeneration. And this is what it looks like at the best image reconstruction that we currently have. So you can see CSF flow into small vessels. Ultimately, we want to couple this with an actuator, and our actuator is ultrasound, and we can do holographic beam steering of ultrasound to generate patterns simultaneously, both in 2D and in 3D. Ultrasound over the last few years has been studied as a modality for stimulating neural activity, for modulating some aspects of neural activity. In some cases, we can also inhibit neural activity locally. So this will basically be a tool for distributed control, again, with access to the full mouse brain. We put these modalities together, optoacoustics and transcranial ultrasound, with fairly high resolution. This is shown here to scale relative to a mouse cortex. So it gives us, it doesn't give cellular resolution, but it gives the type of resolution that allows you to control small areas in the mouse brain. And we can observe that. We can either use fluorescence. We actually see a small dip in fluorescence because of thermal effects. But then after we can model that, capture that full fluorescence dip dynamics. And after we remove this, we actually can see directly the effects of neuromodulation locally around the simulation site. We're doing this now with holograms as well. So to summarize, the mesoscale work, optoacoustic tomography allows us to what we think is a very good path towards imaging multiple different functional signals potentially across the whole mouse brain, together with fluorescence. And we have it allows us, it's still in development, but will allow us to look at other types of functional signals, and we can combine it with ultrasound stimulation as well. And I want to thank the people that did the work, as well as my long-term fantastic collaborators, Dina Rimberg and Daniel Zansky, and of course you for listening. I'll be happy to take some questions.
Thank you very much. Questions for our speaker?
So an interesting thing in terms of pathophysiology that's just lately become realized. So for example, schizophrenia happens around age 20. Why at age 20? Well, children generally have a lot of, their brains are not myelinated, and the myelination process, kind of completes around age 20. And in some people, just things don't get myelinated correctly. And what's the significance of that? The significance is that it introduces delays that are subtle, and that our typical modalities don't pick those up. So one area of interest, for example, is in post-COVID, why the sense of smell is disrupted. And you've pointed out why it could be. It's just that there's some local demyelination that results in delays. So it's a very interesting window that you've opened up.
Thank you for the supportive comment. It's so I think mental illness that arises in adulthood is a very interesting phenomenon. Like you said, there are things that could relate to myelination. You can also ask, why did neurodegenerative diseases really mostly kick up in early advanced age? And part of that is probably a plumbing problem, which has to do with CSF flow and disruptions in the CSF flow as we age. So looking at all these different processes in different preclinical models is indeed very interesting. And part of all of that.
Yeah, thanks. That was really cool. You talked about extending this to humans ultimately. I agree that would be great. I guess one issue is the larger size, and the second is potentially requiring the AAVs. How far off do you think we are in being able to do that?
Yeah, thanks for the question. So first of all, I should highlight that, of course, your lab at Caltech, Leon Wang's lab at Caltech, is a pioneer in doing optoacoustics in humans, and there are some very exciting results that were published last year, of course, in humans that do not have a skull. Once you add the skull in, then you need to go to lower frequencies, in the order of about 0.5 megahertz, wavelengths that are much larger, and basically something more like centimeter type resolution, or maybe a resolution of a few millimeters. Now, you asked about being able to combine that with essentially gene therapy type approaches that can be used for calcium imaging, etc. That's a great question. So first of all, there are some endogenous processes that do exist in our brain that have that are not just hemodynamic, that can potentially be observed. So maybe we don't need that crucially. There are increasingly gene therapy probes that are being used in humans. It's already being done in the eye, clinically approved, and I know that there are several approaches that are going on for doing it beyond. It's definitely not around the corner, but you could imagine either more advanced delivery systems or just better acceptance of safe viral transduction, opening up some of that.
Thanks.
I'm not involved in it.
A question just for my curiosity. So if you study, for example, visual cortex or visual stimulation, you have ways to quantify the stimulation. For example, the brightness and luminosity pattern. But when you study the olfactory system, how do you quantify the smell or the stimulation?
Great question. Thank you. Basically, first of all, of course, there are specific odorants that have been studied a lot, and we understand very well what we as humans understand in them. Of course, mice might have a completely different experience. But I think the best answer to your question is that my collaborator, Dr. Rinberg, has built behavioral schemes in which mice basically can report distances, pairwise distances between different odors. And so basically through that, you can build a full map of the olfactory space of mice. So of course you don't know absolutely what the animal is experiencing, but you can understand basically how different sensory odors or the experience of sensory odors, how they relate to each other, and basically what smells like what and what doesn't. And then because this is done both in mice and in humans, basically those two maps can be matched to each other, and to a certain extent you can do some kind of relational mapping between the sensory experience of mice and that of humans. I hope that answers.
Okay. Thank you.
So the number of questions, I think we can thank all of the speakers in this great session one more time.